a public health emergency of international concern. This is the highest level of public health alert that the World Health Organization can currently issue. After a 2-year interval, the WHO announced again in August 2024 that the Mpox epidemic now constitutes a public health emergency of international concern. The last viruses to be given the same level of alert were Ebola and COVID-19. You have to understand this is the highest level of public health alert that the WHO can issue. Its main characteristic is that the virus can rapidly cross international borders, affecting human health across multiple countries, and having a huge impact on society and the economy. The WHO Director General stated, "Mpox cases have been present in the Congo for more than a decade. During this period, the number of reported cases each year has been steadily increasing, and the number of cases reported so far this year has already surpassed last year's total. There are over 14,000 cases and more than 500 deaths. This virus appears to be spreading mainly through sexual contact, and it has also been detected in countries neighboring the Congo. These are countries that had previously never reported cases of the Mpox virus. Compared to the same period in 2023, Mpox cases reported in Africa this year have increased 1.6-fold. The surge in Mpox cases in the Congo is caused by two subclades of Clade, one of the Mpox virus, namely Clade Ia and Clade Ib. Research conducted by the WHO in the 1980s described the typical characteristics of Mpox Clade I. Described its typical characteristics. Its mortality rate is about 10%, with the majority of deaths occurring in children. Available data from 2024 shows with an overall case fatality rate of 3.6%, and it may continue to spread through multiple transmission routes. This August, Asia also saw its first case of the Mpox Clade Ib variant, which was confirmed by Thailand's Ministry of Public Health on August 22nd of this year during a press conference. Moreover, in China, during the second half of 2023, the number of Clade II infections also reached over 1,100 cases, and most were among men who have sex with men. It has to be said, in just 2 short years, it has spread to so many countries and infected so many people. Coupled with the recent Clade Ib case in Thailand, it's truly worrying. And a crucial step in easing these worries is to fully understand this virus. That's why we need to talk about Mpox. On April 29, 1958, a research institute in Copenhagen was receiving a shipment of lab monkeys just as usual for the production and research of the polio vaccine. The monkey colonies typically consisted of several hundred individuals, mainly cynomolgus and rhesus monkeys. Most of these monkeys were housed in an animal facility containing 15 small compartments. Each compartment, depending on its size, could hold between 25 to 50 monkeys. For now, let's call this Room A, the main animal facility. Sometimes a group of 100 to 150 monkeys would also be housed in a single room in another building, which we'll tentatively call Room B. This building was located about 180 meters away from Room A. This particular batch of cynomolgus monkeys arrived in 1958 on April 29th from Singapore by plane. Upon arrival, the monkeys were first given a prophylactic intramuscular injection of penicillin and then distributed into 3 compartments in the main animal facility. Their overall condition was good, with only a few animal deaths in the following 2 weeks. However, starting on May 15th, due to the increasing mortality rate of Streptococcus pneumonia, to compare the effects of injected and oral antibiotics, and besides the monkeys, who died suddenly during the experiment, the remaining 100 or so were divided into 2 groups, with 50 in each group, placed in 2 different rooms. The first group received 3 consecutive days of intramuscular injections of penicillin and streptomycin. The second group, on the other hand, was fed pellets with added penicillin. Between the 2 groups, no differences were observed, and the mortality rate in both groups gradually decreased at almost the same rate. The animals seemed to have recovered. However, on June 30th, a monkey in the first group began to show a rash similar to smallpox. At this point, the researchers still couldn't figure out what was going on. Soon after, on July 4th, 2 more similar cases appeared. On July 7th, in the first group, another 2 animals had developed a vesicular rash. The last case was discovered on July 14th, apparently already in the late stages of the disease. This outbreak lasted for 2 weeks, involving a total of 6 animals. The remaining animals showed no signs of illness. In the second group and among the monkeys in other rooms, no signs of the disease were observed either. Over the next few months, things seemed to have quieted down. The research institute continued to receive lab monkeys as usual, continuing their vaccine research. That is until November 7th, when researchers once again discovered in Room B that 3 monkeys had developed skin lesions similar to those in the first outbreak. Within the next 3 days, 8 additional cases were observed in the room. When the monkeys were carefully examined a month later, another 12 animals were found to have scattered, healed lesions indicating that among this batch of monkeys, a total of 30% of the animals had been previously infected. According to the researchers' investigation, it was discovered that this group of 223 macaques had arrived by airplane on September 18, 1958. About 100 monkeys were housed in the main animal facility, while the rest were kept in Room B. For a while, right after they arrived, aside from a few fatal cases of pneumococcal infection, all the animals were in good health until November. That's when cases suddenly started appearing in the group. What puzzled the researchers was that the first smallpox-like outbreak happened in the group of monkeys in the main building, while the second outbreak occurred 4 months later in the group of animals in Room B. During the time between these two outbreaks, the monkeys showed absolutely no signs of illness. Right before the outbreak began, by the time the disease fully developed, the clinical sign was a widespread spotty rash. Judging by the scratched appearance of some of the lesions, especially on the animals' backs, the rash appeared to be itchy. The lesions were distributed over the entire torso, tail, face, and limbs, and were particularly dense on the palms of their hands and soles of their feet. These areas also featured larger papules, with contents that were extremely viscous and pus-like. Throughout the course of the disease, the animal's general health didn't seem to be significantly affected. No diseased organs were found during autopsies either. It's worth mentioning that both of these outbreaks were confined to the monkey population. No cases of human infection occurred. There was no doubt about it, the pathogen was out, and it had picked the perfect place to strike. It exposed itself. Faced with the pathogen's provocation, the researchers naturally wouldn't pass up the opportunity. If they discovered something new, winning a small award wasn't out of the question. So they decided to isolate the pathogen. Virus isolation had been attempted on 1 monkey during the first outbreak, and then again on 2 monkeys during the second outbreak. The virus was successfully recovered from all 3 animals, and the researchers confirmed that the same virus was responsible for both outbreaks. They also described this virus in the literature. A virus was isolated from the pustular lesions. In cultures of monkey kidney cells, human amniotic cells, and HeLa cells, the virus produced cytopathic effects. The virus was serologically related to the vaccinia virus and had the typical brick-like appearance of a pox virus. However, unlike smallpox, it could be transmitted via the intracerebral injection route and be continuously passaged in mice, leading to encephalitis, and it produced severe hemorrhagic lesions in the skin of rabbits. In 2-day-old rabbits, it caused a fatal infection, producing typical pathological changes in the liver and kidneys. Finally, the authors also mentioned that attempts to identify the monkey virus as any known pox virus had failed. In other words, humanity had discovered a new type of pox virus. Danish virologist von Magnus and his colleagues were the first to isolate it from infected monkeys and publish their findings. The first paper in this references is the very article he published more than 60 years ago. Produced severe hemorrhagic lesions in the skin of rabbits. In 2-day-old rabbits, it caused a fatal infection in the liver and kidneys, producing typical pathological changes. Finally, the authors also mentioned that attempts to identify the monkey virus as any known pox virus had failed. This meant that humanity had discovered a new type of pox virus. And Pox, the Danish virologist von Magnus and his colleagues were the first to isolate it from infected monkeys and publish their findings. The first paper in the references comes from an article he published over 60 years ago. This is monkeypox, first appearing in the public eye. Over the next 10 years, from monkey colonies in the United States, the Netherlands, and France, multiple monkeypox outbreaks were observed and several isolates were identified. During a monkeypox outbreak at the Rotterdam Zoo, several different primate species showed varying degrees of disease severity. Orangutans were particularly susceptible, dying during the acute viremia stage before any skin lesions appeared. However, in all of these incidents, no cases of human infection occurred. Equateur is a province of the Congo, located in the northwestern part of the country. Basankasu is a town in this province, covering an area of about 20,000 square kilometers. The estimated population is 62,000, and most residents are subsistence farmers living in small villages. The entire region is covered by dense tropical rainforest. The rainy season lasts from February to November, and even during the dry months of December and January, it typically rains 2 to 3 times a week. Major road runs from west to southeast through the Basankusu region. The majority of the population lives along this road and its branches. The main road is blocked by several rivers with no bridges. To reach the region by land from the capital Kinshasa takes at least 7 to 8 days, and Kinshasa in turn has an inextricable link with a host of high-risk viruses like HIV, Ebola, and others. The town of Basankusu has a 150-bed government hospital, staffed by 1 doctor and some nursing and technical personnel. They usually conduct routine smallpox vaccinations here. When infectious cases are discovered, the information from here is also reported to the province. In 1969, at the Basan-Kusu Hospital, several suspected smallpox cases were handled, but not a single one was confirmed. In 1972, smallpox cases were reported. An investigation found that one of them was a typical case of chickenpox, which had been mistakenly reported as smallpox. As for the other patient, a throat virus was isolated from him. This patient was named I, a 9-month-old boy who developed a fever on August 22, 1970. 2 days later, a rash appeared. He was admitted to Basan-Kusu Hospital on September 1st. Examination records showed the lesions were hemorrhagic, and although the rash showed the typical outward-spreading distribution of smallpox, it lasted for about 2 weeks. During the scabbing stage, the patient developed an ear infection, as well as enlarged, painful lymph nodes in his neck. Fortunately, the little boy recovered from the illness, even though he had not been vaccinated against smallpox. Unfortunately, just as he was about to be discharged, he contracted measles and died 6 days later. A specimen from the patient was later sent to a World Health Organization, Smallpox Research Center, and a virus similar to the monkeypox virus was isolated. Researchers confirmed that this was the first case discovered in 1970 of a human infection caused by the monkeypox virus. A later epidemiological investigation revealed that AI was the youngest child in the LD family. LD had 2 wives, 11 children, 1 daughter-in-law, and 2 grandchildren. The family members had, for many years, lived in the village of Bokenda. One of LD's wives, who was also the patient's mother, BM, left the village of Bokenda on August 18th with the patient and Hunt's 4 other children to travel to the village of Balanda, 115 kilometers away, to visit his maternal grandmother. The family arrived in Balanda village on August 20th. 2 days later, the patient developed a fever. Before their departure, AI and his mother had been staying in the large village of Bokenda. Family members denied having any contact with anyone who had a smallpox-like rash. Investigators also found no other cases in the village. Ai's father said that the family occasionally ate monkey meat, considering it a delicacy. However, his parents weren't sure if AI had eaten monkey meat in the months before he got sick. They also couldn't confirm if AI had direct contact with any killed monkeys during that time. Notably, because monkeys are abundant in the area, they have been consistently hunted in the region as a source of both food and income. The monkey meat is usually eaten in stews or smoked and sold in local markets. The local residents hunt monkeys extensively. Sometimes when a hunter brings back some monkeys he's killed, you'll see children immediately gather around them and start playing. With that in mind, the question of how monkeypox found its way to humans seems to have an answer, even if it's just speculation. Several studies in Africa have reported that the monkeypox virus is most likely transmitted through contact with the bodily fluids of infected animals like monkeys. For example, getting bitten by an infected animal while hunting or coming into direct contact with the virus while transporting, butchering, or preparing the infected animal for food can lead to infection, which then spreads among the human population. For a very long time, monkeypox was something you really only found in Africa. Since 1970-10, human monkeypox cases were reported in an African country. From 1996 to 1997, the Congo in fact had a massive outbreak. A total of 511 cases were reported, with 5 deaths. From 2001 to 2002, the Congo confirmed another 485 cases, with 25 deaths. Then in 2003, monkeypox finally broke out of Africa and achieved its own American dream. This was also the first time monkeypox had ever appeared outside of Africa. And this whole story, well, it all starts with a group of adorable prairie dogs. By the way, they have a pretty cool official name, the prairie dog. For the sake of cuteness, we'll just stick to calling them that. On May 24, 2003, in Wisconsin, the Department of Public Health was notified that a 3-year-old girl, after being bitten by a prairie dog on May 13th, was hospitalized with a fever. This prairie dog had been purchased at an animal market on May 11th. It became ill on May 13th and died on May 20th. Its swollen lymph nodes were sent to a lab for bacterial culture. At first, tularemia or plague were suspected. It was ultimately identified as Acinetobacter and was considered a contaminant. With no similar illnesses reported, the case appeared to be an isolated incident. On June 2nd, the local health department received another report. A man had fallen ill. The man lived in southeastern Wisconsin and was also an exotic animal dealer. On May 18th, he was bitten and scratched by a prairie dog. 7 days later, he developed a fever, chills, and swollen lymph nodes. Later, an investigation revealed that it was him who had sold 2 prairie dogs to the first patient's family, and the fact that his prairie dog got sick was possibly due to contact with another sick was related to Gambian giant rats. During this outbreak, the first cases evaluated in Wisconsin were 5 men and 6 women, with ages ranging from 3 to 43. All patients reported having a febrile illness and direct contact with sick prairie dogs before the rash appeared. The virus was later identified as monkeypox through testing. In 2017 and 2018, Nigeria reported the largest monkeypox outbreak in the country's history, with a total of 122 reported cases. In 2022, monkeypox had its moment in the spotlight again, making the WHO break out in a cold sweat that year. And they declared that monkeypox had become a public health emergency of international concern. That year in Africa, a total of 5 countries reported monkeypox, cases with over 1,300 cases and 68 deaths. Outside of Africa, the UK, Spain, Portugal, the United States, Canada, and 23 other countries also reported monkeypox outbreaks, with a total of 257 confirmed cases and about 120 suspected cases. The outbreaks in multiple countries showed that the monkeypox virus had spread from person to person, and it was primarily clustered outbreaks among men who have sex with men. So what about the monkeypox situation here in China? I did a quick search and made a simple summary. According to the China C.D. So Weekly, our country's first monkeypox case was discovered in Chongqing in September 2022. A 29-year-old Chinese salesperson visited Germany on September 2nd. On September 2nd in Berlin, he engaged in MSM activity, that is, men having sex with men. He then traveled to Spain and returned to Chongqing on September 14th. Was later diagnosed in Chongqing with monkeypox Clade Ib. Because I couldn't find comprehensive, authoritative national data, I've summarized the Mpox case situation released by the Chinese CDC from August to December 2023. The Chinese CDC released on Mpox cases from August 1st, 2023 to December 31st, mainland China, confirmed a total of 1,115 cases. Of these, Guangdong had the highest proportion from August to November. I couldn't find specific provincial data for December. The main group infected in this outbreak are men who have sex with men, accounting for over 90% of cases. Lunyu et al. Published a research paper in Nature on the evolutionary trajectory of Clade Ib. Of the evolutionary trajectory of Clade II, which also mentioned the transmission risk among MSM. The study included 92 Mpox cases. All patients were male, with a median age of 30. Of these, 95.7% engaged in male-to-male sexual behavior, and 56.5% of the patients were also co-infected with HIV. Then there's the situation this year, which I mentioned at the beginning. In the first 8 months or so of 2024, the number of global Mpox cases has already reached over 17,000, with more than 500 deaths. To have accumulated so many cases in just a few short months. The terrifying part is precisely this exponential growth. What's more, the first cases of Clade Ib also appeared in Asia in August. The English abbreviation for the monkeypox virus is MPX5 or MPV. The World Health Organization now recommends using the new preferred term, Mpox, as a synonym for monkeypox. The Mpox virus is a double-stranded DNA virus with a full length of about 197 kilobases. It belongs to the orthopox virus genus of the Poxviridae family, and in China it has been classified as a Class B infectious disease. Infectious disease management. The virus is very easy to cultivate artificially, which is why some scientists are concerned that it could potentially be exploited for development as a biological weapon. The morphology of the monkeypox virus is consistent with other orthopox viruses. It has a rounded brisk-like or oval shape, measuring 200 by 250 nanometers, and is surrounded by a 30 nanometer outer membrane. The monkeypox virus, along with the smallpox virus, the vaccinia virus, and the cowpox virus, are the 4 types of orthopox viruses that are pathogenic to humans. Their antigenic properties are basically the same, and they share cross-immunity with one another. This is why getting the smallpox vaccine can usually provide some protection against monkeypox. But since 1980, when smallpox vaccinations were stopped, herd immunity has gradually declined, giving monkeypox an opportunity to break out. To date, humans have discovered 2 evolutionary clades of the monkeypox virus, Clade I and Clade II. As for Clade I, you might also see it referred to in other official sources, by names like the Central African Clade or the Congo Basin Clade. These names more specifically indicate its geographical distribution. Clade I also has subclades, which are IA and IB. Meanwhile, Clade II, also called the West African Clade, also includes 2 subclades, IIA and IIB. The genomic difference between these two genetic clades is less than 1%. However, it's this less than 1% difference that leads to a huge gap in their case fatality rates. China's technical guidelines for monkeypox prevention and control state that the case fatality rate for patients with Clade II monkeypox is about 3.6%. The case fatality rate for patients with Clade I monkeypox, however, can be as high as 10.6%. Historically, the clinical presentation caused by Clade I all showed more severe characteristics than Clade II. Studies have found that the Mpox virus genome contains multiple genes that code for proteins which regulate the immune response. These proteins include the Mpox virus complement inhibitor, which is believed to be a factor in the enhanced pathogenicity of Clade I. In Clade II, however, this protein does not exist. The strain that caused the 2022 global Mpox outbreak in non-endemic regions was Clade Ib. Since September of 2022, when China reported its first imported Mpox case, the virus sequences sent to the Chinese CDC from various regions have all been from the Clade Ib branch. However, starting this year, in addition to the initial Clade Ib, a branch that mainly affects children, the Clade Ib branch has also begun to spread in the Congo. Although this strain was first identified in 2024, it is estimated to have already appeared around September of 2023. The outbreak in the Congo caused by the Clade Ib branch primarily affects adults. And what's worrying is this evolving branch Clade Ib is also being found outside of Africa, including cases in Thailand, and it is spreading rapidly. It is estimated that the epidemic is primarily, but not exclusively, sustained through transmission related to sexual contact, and is expanding within networks associated with commercial sex and sex workers. The Mpox virus is resistant to dryness and low temperatures. It can survive for several months on surfaces like scab soil clothing and bedding. The virus is sensitive to heat. Heating it to 56 degrees Celsius for 30 minutes or 60 degrees Celsius for 10 minutes can inactivate it. 75% ethanol, chlorine-based disinfectants, peracetic acid, and other common disinfectants, as well as ultraviolet light, can effectively inactivate the virus. Is sensitive to hydrogen peroxide, sodium hypochlorite, and formaldehyde. When a virus invades the body, this is usually how it makes you sick. The Mpox virus can enter the human body through the respiratory tract or the skin. In the respiratory tract, the virus can infect the epithelial cells of the airway. After being introduced to the skin, the virus infects keratinocytes and fibroblasts. Immune cells residing in the skin are also targeted. In both cases, the virus then enters the lymphatic system and spreads through it. A common feature of human Mpox is swollen lymph nodes. After the virus spreads through the lymphoid tissue, the Mpox virus may target other major organs like the spleen and liver. The subsequent viremia may allow the virus to spread further to distant organs, like the skin and reproductive organs. Infection of the skin and mucous membranes leads to the appearance of infectious pustules and ulcers, and releases infectious viral particles into the saliva. This forms the basis for the aerosol transmission of the Mpox virus. However, it's still unclear how the virus reaches the upper layers of the skin, which lack blood and lymphatic vessels. Some say that once the virus reaches the upper layers of the skin, it marks the beginning of the skin infection and lesions. Generally speaking, Mpox is a self-limiting disease. The severity of the infection can be influenced by many factors, such as different viral strains and individuals' immune status, and any potential complications that may arise. Severe cases of Mpox can lead to hemorrhagic disease, necrotic diseases, obstructive diseases, complications like inflammation of vital organs and sepsis. Furthermore, individuals with weakened immune systems, including children, the elderly, and the immunocompromised such as people with HIV or those taking immunosuppressant drugs, are more likely to experience these severe symptoms. What's more, individuals with weakened immune systems are more likely to help the Mpox virus evolve, allowing it to become better adapted. Now let's talk about the transmission routes of Mpox. To give a more complete picture, the virus can enter the human body through mucous membranes and broken skin. It's mainly transmitted through contact with an infected animal's respiratory secretions, fluid from its sores, blood and other body fluids, or by being bitten or scratched by an infected animal. Between people, it's mainly spread through close contact. Close contact includes skin-to-skin contact like touching or sexual activity, and mouth-to-mouth or mouth-to-skin contact. It can also be spread through respiratory droplets during prolonged close-range contact. Touching items contaminated with the virus can also lead to infection, such as clothing or bed sheets used by a patient. The virus can also be transmitted through the placenta from a pregnant person to their fetus. A person is contagious from the moment symptoms appear until their sores have healed and the scabs have fallen off. Generally speaking, the incubation period for Mpox is between 5 to 21 days, but it's most often around 6 to 13 days. Early on, you might experience initial symptoms like fever chills, headache, drowsiness, fatigue, back pain, or muscle aches. 90% of patients develop noticeably swollen lymph nodes. A rash appears 1 to 3 days after the initial symptoms. It usually starts on the face and gradually spreads to the limbs, palms of the hands, and soles of the feet. The rash goes through stages from flat spots to raised bumps, then to blisters and scabbing. Rashes in different forms can be present at the same time. Vesicles and pustules are mostly spherical and can be accompanied by significant itching and pain. The number of vesicles can range from a few to several thousand and can affect the oral mucosa, digestive tract, genitals, conjunctiva, and cornea. Most monkeypox patients recover within 2 to 4 weeks. Some patients may experience complications, including secondary bacterial infections of the skin lesions, vomiting, and diarrhea, causing severe dehydration, bronchopneumonia, encephalitis, and corneal infections, among others. As for those who are not infected, there are preventive measures you can take, such as frequently washing your hands with soap and water or an alcohol-based hand sanitizer, especially after suspected or actual exposure. If you have sex, using a condom can reduce the risk. Based on the transmission routes of the monkeypox virus, take protective measures in high-risk areas. Although there is a vaccine for monkeypox, the current vaccine supply is quite limited. There are mainly 2 vaccines, the MVA-BN vaccine and the ACAM2000 vaccine, as well as one that was just approved for clinical trials in China on September 9th. The MVA strain monkeypox vaccine, independently developed by Sinopharm Group, which is a live attenuated vaccine. Coincidentally, September 9th was also the final day for finalizing this script. In 2022, the World Health Organization studied these vaccines in different populations to assess the safety for subjects at high risk of exposure to the monkeypox virus, as well as their immunogenicity, potency, and effectiveness. Friends who want to learn more can check out the article about vaccines in the references section. To date, there is no proven effective antiviral treatment for monkeypox. And finally, to satisfy everyone's curiosity, let's talk about where this virus came from this time, or rather, which little critter is responsible. According to an article in The Lancet, the host of the monkeypox virus is still unclear. But serological surveys indicate that African rodents, such as squirrels, non-human primates, and rats, may be its natural hosts. One species of squirrel showed a 24% monkeypox virus seropositivity rate, which is significantly higher than other animals. Even the Gambian giant rat only had a positivity rate of 16%. Several national epidemiological studies have also shown that rodents living in agricultural areas are the main candidates for transmitting the virus to nearby residents. This means that both serological and epidemiological studies point to African rodents as the source. Of course, its intermediate hosts like monkeys and apes are also significant sources of infection. In 1979, humanity declared that smallpox had been completely eradicated. But little did we know that not long after, another formidable member of the pox virus family, monkeypox, after lurking quietly for many years, would suddenly burst onto the scene this year with increasing ferocity. Perhaps the infinite war between humans and viruses will go on forever, and what we ordinary people can do is to build our awareness, stay vigilant, and take preventive measures. All right, that's all for this video. If you found this helpful, please give me a like to show your support. Thank you, everyone. If you like our videos, remember to follow. I'm Yuntangguo. See you next time.